So things that are going to happen now, what were some of the ideas that you were thinking about? Okay, don't everybody jump in under this at one time.
So do we know enough about the parasite? Have we got a good handle on these genotypes?
I'd say the only thing that I can think of is if the dams are removed, then like how are they going to evolve when you see the upper basin genotypes in the lower basin fish?
When all the fish start moving around and transporting the parasite and changing the
Well, and I think
population.
we have a, I mean, if we assume that when the dams went in, there was evolution there for adaptation to the different hosts in the different systems, then when the dams comes out, you would think there'd be evolution at that point too, to either be a generalist parasite and kind of affect everything.
See, this is perfect.
Specialist.
Well, and indeed that's where we might start to see competition between the different strains, because suddenly they don't have their own little fish population to themselves. All the fish are moving around.
It's a big
And
selection
there's
pressure.
the idea that the different fish species are also genetically different. The populations of red band are different, so then maybe they have their own particular subgenotypes.
Which is not something we've even really looked at yet. There's that level of detail that I guess we haven't needed to go there yet.
Well, I would follow on from what Charlene and Stephen have said, and that I'd like to know exactly how many genotypes there are out there. And I would love it if we could sequence the whole genome of Ceratomexa, because
get a better handle.
I'd like to find a gene that suits my objective of being able to design a diagnostic method where we can see exactly which strains are in the water and how much of each, and we can just do one test and we can find out all that information relatively quickly. That would be great.
And
very, very useful, I'm
instantly
sure, to all of
assess
us.
the risk to the fish population at that point.
Yes, management implications.
Yes.
It'd be useful to lots of us.
And that would be useful for Matt's project.
Yeah, so we don't know exactly from all these different other species, like white trout are kind of something I've been well used to getting an example of, or whitefish, not white trout. And then like coastal cutthroat, there's still some species out there that we haven't even tested yet.
Yeah, exactly.
And they're actually, endangered in some ways, so it's harder to get samples from them, but that might be part of the problem, so
And I think the
hopefully we can
evidence
try
we
it
have
out.
to date is, I bet if we start looking at different trout and salmon species, we're going to find new genotypes of this type of parasite. Maybe new species, maybe new genotypes. That's sort of a bit of a flexible boundary there, I think, how we define it.
I think one thing we need to work on that Michelle and Julie are answering is the polychaete habitat and polychaete numbers, and that's the huge part that we don't really know anything about. We need to understand what they're doing in the lower system right now, and then we need to try and guess or predict what they're going to be doing once the dams come out, if the dams come out, and see where they overlap. I think that's the big, we understand the fish pretty well. We kind of have an idea of what they're doing. And the genotype thing is interesting, but in terms of the river itself, the polychaete's the big black box that we don't know anything about really.
Well, it's the big contributor of spores to the fish.
Yeah, and it's been there, it's always been there, it's going to be there, but we just need to figure out what we can do about it.
Yeah, and to build on that, we have no idea what the effects of dam removal will be on the invertebrate community, like the benthic community that maybe is filter feeding and therefore affecting the dietary supply to the polychaetes or somehow competing with them for spores. We don't know anything about the effects of dam removal on that at all.
Do we expect any direct effects from removing the dams in terms of scouring flows and things? I mean, that's sort of your quote-unquote natural system, but is removing Iron Gate Dam and I guess some of those
If
other...
anything, it could almost have the exact opposite effect. I mean, the 2006 data set that we're looking at right now was collected after a huge flow year. There was a lot of stormwater runoff and snowpack runoff, I guess. And so you would think that would have had a huge scouring flow effect, and the densities of polychaetes in those samples are way higher than what Michelle and I are seeing this year. So we just don't know enough about it yet to say. It certainly could, but I don't know.
Right, I think the questions of hydrology effects on this life cycle are a real outlier here, and if you look at other systems that are perhaps not as managed as this, the dynamics of the disease are quite a bit different.
Are they better?
It's hard to say whether or not they're better, they're just not as well studied. And sometimes disease is a problem because we know to look for it.
And that's certainly a problem with any of these more and more sensitive diagnostic techniques we have, the more sensitive we can find parasites everywhere if we look sensitive. But the question is it a problem? I think that's where it really comes back to what you're
Is it
sort of
biologically
saying.
significant?
Looking at the hosts, and is it affecting the fish populations? Is
Yeah.
it affecting the polychaete populations?
Yeah.
Can we think of some bigger ideas outside of the
Like
system?
climate change, should we start talking about it without Wei Chao?
Sure.
Okay, yeah, when temperatures are going to be changing, in the climate, run timing has kind of been a big idea. If stocks start coming up earlier or later, and the parasites are released, on top of these fish that when they're more susceptible, it might just start cutting down their life cycle before they can spawn, or even juveniles trying to out-migrate. If they're getting later on in the year and the water temperatures are too warm, they won't be able to survive that out-migration.
Is there even a chance that it might help the fish populations if they migrate at different times? If currently we've got this sort of balance between when the parasites are in the water and when the fish are coming back, if the fish start to change their behaviour because of increasing water temperature, might they have an advantage for a couple of years before
I think that's gonna depend on how the polychaetes then respond to changing temperature as well. So with the sampling that we did this year, we saw it looked like there was a correlation between population size and the temperature increase that happened. It was a very late spring, and suddenly the temperature spiked, and we saw the populations explode. And so, if the polychaetes their life cycle is very tied to temperature as well, then a shift in temperature regime might also shift their life cycle. So, and then that would affect then the fish in a secondary manner.
And I think temperature, Adam, correct me if I'm wrong, worked in the fish's favour this year.
This year, yeah.
Because we had lower levels of parasite in the water, but because of the temperature differences, we had lower temperature this year. The effects, the impact on the fish was not as severe.
Yeah, so temperature could actually have a role in parasite development. Like, it may take a certain amount of degree days or temperature units to actually get the parasite to develop to an infectious stage.
Are we seeing any current changes arguably because of climate change? Are we seeing run times shift from historical records?
The run times in the climate, they're so hatchery dominated that historically the salmon run was a spring run, so the adults were coming back earlier. And I think that's kind of, now they're selecting for a fall run, so now the adults are coming back at this point in time. So.
It's completely irrespective of climate, it's just the hatchery time.
It's just a hatchery
time.
driving
Yeah.
it. So.
And can we say there's been a trend anyway? I mean, I've been here for several years now, and every year seems to be unique.
On the Klamath,
Yes.
Yeah.
So I don't know if we can see some overall trend and what time frame, what time scale we're working with, but every year seems to be a bit different.
Yeah.
It's kept me on my toes at any rate.
Yeah, that's huge too. This idea of collecting data and seeing patterns on different time scales, because the variation that we're seeing year to year here, which seems huge, when you step back to a certain time step, instead of going like this, you're seeing suddenly those fluctuations become periodic as well, or maybe have a trend. And so for us, we've got data on the Klamath for how many years have we been doing?
About 8 years.
About 8 years, which is just a drop in the bucket, especially if you're talking about any sorts of questions relating to climate change, which is
Or evolution.
hundreds and thousands, and evolution, which is
Yes.
hundreds and thousands of years. So,
But I'd also emphasise the point of the importance of establishing index sites and going back year after year to establish these patterns of the data. I think that's a significant thing that we have to make sure we are going back year after year, redoing it, watching these trends as they evolve over time.
Yes, it's certainly a lot different working in the field than working in the laboratory, where you can narrow down your variables. You can test a single variable at a time. When you take things out into the field, you don't even know what your variables are some of the time. You know certain variables, but they may not be the ones that are actually affecting the outcome of your study.
And then they're different year to year.
Yes, yes.
or you may find a correlation, but in fact, the things you're correlating are proxies for the actual underlying cause, which
Exactly,
then,
you don't understand
so
what the
you-
underlying cause was, you're
Yeah.
just drawing a false conclusion,
You're just seeing
potentially.
the pattern as opposed to the process.
This is a totally different topic for Big Idea, but as the resident describer of parasites, do you have a sense of systems that are more controlled, like the Klamath, that are dammed? Do you think there's lower parasite biodiversity? in systems like that relative to something that's more natural where things are maybe not as out of balance?
I'm not sure it comes down to directly whether you can say this is a highly managed system, therefore the diversity is one way or the other.
Mm-hmm.
I suspect what it comes down to is the diversity of hosts that are available to these parasites, especially with what we're working with these myxozoans, where they require a specific, they probably require a specific vertebrate host and a specific invertebrate host.
Mm-hmm.
To get a whole lot of different myxozoan species, you're going to need a lot of these pairs present. You're not going to just need a variety of fish species present. You're going to need a variety of that benthic
Mm-hmm.
worm fauna. So I don't know. I would argue that if a managed environment had a sufficient range of hosts, it could support a sufficient range of parasites. It might not be what was there originally in a natural system, but
Mm-hmm.
I don't think there'd be a direct relationship between this more managed river has more or less
Biodiversity.
biodiversity
But I think
parasites.
in that respect that myxozoans are a very good indicator of the health of a system,
Right.
because you need at least two hosts, and ultimately with the parasite and those two hosts you've got three groups of organisms having to be represented. So if you've got a myxozoan present, you need to have three different groups, from a myxozoan to likely an annelid
Yes.
host to a vertebrate. So it really, I think, does reflect on what's going on in the bigger
Well, and I
picture
think
of
that's
things.
why see Shasta is such a good organism to study, because it is so dependent on its two hosts.
Mhm.
I think if we're going to be monitoring the Klamath River in the longer term, then we've chosen a good organism.
What's fascinating to me is that these life cycles are so exquisitely adapted. You can only have a salmonid host, you can only have Manianchia speciosa as the worm host. And what I am really interested in the evolution of this life cycle. And that goes back to if you look at the life histories of these organisms, you're looking at a salmonid host that spends part of its life cycle in saltwater, and a polychaete worm, which is traditionally known as a saltwater species.
And there are so few freshwater polychaetes available.
Yes, there are only about 11 freshwater polychaetes that we know of. Ceratomyxa Shasta is only one of only, 5 freshwater ceratomyxas. Most ceratomyxas are marine species. So this is really a bridging organism. It bridges the freshwater myxozoans and the saltwater myxozoans. And that kind of led into some ideas that you had about Manianchia distribution.
No, I just think it's really interesting. It's a really interesting organism in that, like you said, there are so few freshwater, and it's also, I think, wild how one of its closest relatives lives in Siberia, Manianchia bicolensis, and it brings to mind these ideas of how did this distribution come about, and how did this evolution come about, and there's theories about glacial lakes and that sort of thing, and how did they get inland initially, and then how did they eventually, so they had to migrate inland, and then they had to adapt to freshwater then, which I just think is really super interesting. So, and that's on a huge time scale again, that's millions, thousands, millions of years. Something like
Well, I
that.
think polychaete worms have been around forever. I think both fish and polychaetes have been around 500 million years at least, so there's a lot of time there for these things to evolve.
Mhm.
They don't move very fast though.
No, I mean, what are we assuming? They're just spreading by
They're terrible
making
dispersers.
little...
They're
Yeah.
Surely they move downstream easily if they're
Yeah.
dislodged or whatever, but I don't know how they get upstream.
Yeah.
They walk very slowly, and it maybe happens over millennia, but
Tentacles.
yeah.
Build their colonies boulder by boulder,
Exactly,
working their way
exactly.
upriver. I don't know.
That's a great question, and this has probably been answered by someone, but a lot of aquatic invertebrate species have a flight stage, so they emerge from the water and then they move upstream, and that's how they keep from just getting flushed down out to the ocean. And this organism,
How does it replenish?
it doesn't have that type of a stage, so how does it keep from shifting eventually downstream?
Well, and I wonder if that's kind of a remnant of its marine origin, that you didn't need that kind of a life cycle. And when you, again, looking at this life cycle, I guess we have assumed, or are assuming, that the annelid is the natural host for this, and the fish probably came as a secondary host to move the parasite around.
Yeah,
That
I
may
think
not be
that's
true, but that's an
I
assumption.
think that's the position for myxozoans
Yes.
in general, that
Yes.
the invertebrates are the original host.
Mhm.
The invertebrates have been around longer than the vertebrates, and this is presumably an invertebrate host.
And this is, the parasite itself is likely derived from the Cnidaria, so it's, again, a saltwater organism. So there are a lot of connections here that I think would, nobody's really sat down and tried to piece them all together with what we know now.
Yeah.
As a sum-up question, how about, what are people thinking about interrupting the cycle? Is that a bad question?
No, it's everybody.
It's his course,
Yeah.
project
Mhm.
thing.
It's Adam's job.
It's his focus.
For interrupting the cycle, I think a lot of it is going to have to be focused in on the polychaetes somehow, just because, again, we don't know a lot about them, but there's not a lot we can do with the fish. People want to keep the fish, they want to catch fish, so that's kind of a given that it's going to be around no matter what. So trying to get more information on the polychaetes and seeing how, I guess, different different flows, I think, is one thing that we can actually manipulate now, potentially with the dams, if we can kind of come up with a plan maybe. But seeing how different velocities may or may not affect the transmission of the parasite, I think that could be key. Just because the parasite can't really control where it goes, it's going to go with the water no matter what. So if we can try and manipulate that a little bit, that might be one pretty easily feasible, at least, experiment in practice, since we can control the water to some degree in certain areas.
Are there any thoughts on putting, inoculating the fish or immunizing the fish in some way to the parasite? Has there been any work done?
Everything I've seen or read, it says they haven't figured out how to immunize against it yet. But
Well, it's an idea I've had, but I haven't really looked at it that much. But we're testing that right now in the laboratory, so we'll see what happens.
What exactly are we testing, Shelley?
Protective immunity in the fish. Exposure to one genotype that may not be as virulent, and then exposure to a second that is potentially more virulent.
With the hope that
The fish will have some sort of resistance at that point, and will either have reduced myxospore numbers or will have better survivability.
So you could maybe expose fish to one genotype while it's still in the hatchery before you release them into the river, where they could get exposed to a more virulent genotype, I guess.
Could. I don't know how well that would be received in the hatchery practice.
It saves the fish.
And it depends on how fast the parasite evolves. Like if you've got an evolution timescale of 5 years before you have a new genotype, then that seems really risky. So we would have to have a better idea of how fast they're evolving before we wanted to implement something like that.
And I think one of the things when you're working with pathogens that are problems in the wild rather than just in the hatcheries, you have to deal with how you would manage it for the wild populations as well as the hatchery populations. And so we do have options on how we might be able to do things with hatchery populations, just like, protective immunity might be an option with the hatchery population. Changing release timing so that those fish are not going through the river at the time when the pathogen levels are at their highest might be an option for hatchery populations. You're much more restricted on what you could do with wild populations. And I think that's where, we are restricted to things like controlling the polychaete populations.
Unless you see a significant reduction in myxospore numbers by immunity.
Mhm.
Then if you saw a significant reduction in myxospore numbers, you would see hopefully reduced parasite densities overall, but it would depend on what those levels were.
Mhm. Right, I guess it's possible that if hatchery populations were coming back with a much lower infection rate, that might
10%.
overall reduce the levels in the system.
once Charlene has that data, can you input those numbers into your model and actually see if that's a possible scenario?
Yeah, we have, there is a myxospore production component in the model from salmon. One of the questions now is it juveniles or adults that are the main contributors? Because looked at juveniles from one of my experiments and they can produce millions of myxospores or hundreds of thousands of myxospores. So it's not insignificant in the terms of what's going on.
Yeah.
But
When I think it
Most
only
likely
takes
it
about
is.
100 to
Yeah.
affect a loan.
Okay. And most likely it is the adults that's bringing it back up the river, and if they are producing less, then it might stop the cycle a little bit or dampen it a little bit. So.
Okay. Anyone want to say anything else?
Cold or wet tooth.